Strasbourg in France, and the group of Nobel laureates Jean-Pierre Sauvage. And after that, he moved to the Netherlands as a postdoc in the groups of Gerard van Coten at Utrecht. He also worked with Jan Rietzig at Leiden and Antoinette Kilian and Bert Klein-Kepnik also at Utrecht. Between 2009 and 2014, he completed a tenure track position in inorganic chemistry at Leiden University, where he was tenured in 2015 and became a full professor in 2020. Now, Professor Bonnet has been awarded several prestigious grants, including a starting grant from the European Research Council in 2013, and 3 young investigator grants in 2008, 12, and 19 from the Dutch Organization for Scientific Research. Since 2015, he is a fellow of the Young Academy of Europe and became a board member thereof in 2017. Now, Sylvestre's expertise lies at the crossing point between bioinorganic chemistry, photochemistry, and lipid membranes. His current research interests are anti-cancer, photoactivated chemotherapy, supramolecular photocatalysis, and conversion. And the presentation today is titled Photoactivated chemotherapy based on transition metals towards the treatment of hypoxic tumors. So please welcome Professor Bonnet.
Thanks a lot, Siga. I don't know. Yes, I will share my screen. So can you see everything? Working
Yes.
properly? Yes?
Yes. Yes.
Okay, thanks a lot. Well, many thanks to you, Siga, for your introduction, of course, but also for the invitation to speak in this conference. I was really looking forward to go to Iceland, as you probably expect, because I've never been there. And well, we have to stay home to face the digital loneliness, but I will try to entertain you a little bit on the metal-based anti-cancer drugs and try to explain you the research that we have been doing in the last, let's say, decade or so when I started on this topic. So actually, the research I'm doing now obviously has some links with the research I did during my PhD, although it's quite different as well. So I did my PhD on molecular machines controlled by light. And I learned there that some transition metal compounds, in particular those based on ruthenium, there are a couple of others, are capable of doing very interesting reactions, which are quite easy on paper. You take a ruthenium complex of a certain kind. So this one has 3 nitrogen ligands here, 2 nitrogen here on the BP. So it's called a polypyridyl ruthenium complex, and it's also bound to a sulfur ligand in that particular case. And when you shine light, you very selectively kick out the Thi ether, which becomes free. And 1 molecule from the solvent, so you can do this in water, is binding on the ruthenium. So this is called the photosubstitution reaction. It's quite amazing because it's so selective. There is no, well, quite often there is no other photoreaction going on. The nice thing is that when the ligand changes, you change the coordination sphere of the metal. So the color of the complex changes, which allows you to do all kind of stuff. First, you can see with your eyes that the reaction has happened, which is not the case of all reactions. And secondly, obviously, you can use UV-Vis to do kinetics, to do even thermodynamics sometimes, and to study all these reactions. So this is an easy reaction. Here I show the blue and the green light sources, the spectra of the light sources we are now using actually to trigger these reactions. So the absorption bands of this particular complex is typically has maximum around 450. So anywhere between 450 and 550, let's say. And because this is in the visible region that we came into this idea of phototherapy. So this is a photochemical reaction. Now, when you go from a photochemical reaction to the idea of a light-activated drug, of Of course, on paper, it's quite easy to do. So what is a light-activated anti-cancer drug? Well, you say, okay, imagine the initial molecule, you put it in a syringe, and you inject into a patient who has cancer, for example, here in the lungs. So when you put a drug in a person, usually the drug goes everywhere. Obviously, you do some targeting, and it goes, if you do it properly, will do a bit more in the tumor than in the rest, but there is always some of the molecule going in the rest. So if it's directly biologically active, you will have a lot of side effects because the active drug is killing all kind of cells that are not the cancer cells you're targeting. But if your molecule is not active or much less active in the dark than after light irradiation, then all these orange halo over there is not going to do too much. What you do then is you take a source of light and you shine light specifically onto the tumor in vivo, inside the patient. I'm going to show you how this can be done. By doing so, you trigger the photoreaction in the patient and you transform a compound into another compound. And if this is well controlled, then the other compound is more cytotoxic than the first one, and is going to kill the tumor just because the light has been shown on the tumor specifically, while the rest of the molecule is not doing anything, is not doing too much. And here as well, in a perfect world, the patient is healed without side effects or with very reduced side effects compared to chemotherapy. So this was chemistry or biology on paper. Now, how does it look like in real life? I'm very happy to have this photo from Professor Teresa Bush from the University of Pennsylvania. She gave it to me a couple of years ago because it shows so well what phototherapy actually is in real life. So what you can see on this photo is a patient here. He was operated from a brain tumor. You see here the robot that has been used for the operation. And what you see is that in a cavity of the brain tumor that's been removed, people want to clean the cavity and remove the remaining cancer cells. And for this, they do phototherapy. And you see there is an optical fiber going onto the table. On the table is a laser, shining light. So you really bring the light into the patient. What is also well represented here are the doctors who are in the back of the room. The room is dark, or let's say medium dark. And the doctors are simply waiting for some time, let's say 15 minutes, typically not too long, because you don't want the operation to take too much time, but typically 15 to 30 minutes, until the drug has been irradiated enough to make its action. So this is really occurring in the clinics already. There are many clinical trials. The chemistry at the basis of this, well, the clinically applied phototherapy against cancer is called photodynamic therapy. Probably a lot of you know about it, but I wanted to remind this. So here the molecule is called a photosensitizer. It can be a transition metal complex, but it doesn't have to be. Many are porphyrin-based molecules. The light you shine onto the tumor tissue gets absorbed by the photosensitizer, and you have a form of energy transfer. It can be electron transfer as well to oxygen, which is present in the tumor tissues. You generate singlet oxygen, for example, and this is actually the cytotoxic species. So for PDT, you really need light, you need a compound to absorb the light, and you need oxygen. This has been approved first for esophageal cancer, and you can see here how amazing PDT can work. So this is esophageal tumor obstructing the esophagus before the PDT treatment. So after the PDT treatment, you see the tumor tissue is necrotic. The body has to clean away all the dead cancer cells. And 1 month after PDT, the esophageal is not obstructed anymore, and the tumor is gone. So I'm going to try to show you new chemistry to help with the problem of PDT, but I certainly don't want people to come out of my lecture thinking PDT doesn't work. This is why I show this first. PDT really can work amazingly, and I think it's important to realize. And another thing is important to realize is most of the time, PDT is a technique where you use the oxygen in the tissue to generate the anti-cancer effect. So how do you shine light into patients? Well, I'm not a doctor, so I'm not developing these things, but I need you to realize that they exist. So this is a device, well, maybe most of you think about, let's say, this kind of very easy light application. If you would have a skin tumor, you take a lamp, well, you take the drug, you put it in a gel, the gel on the tumor, you wait for some time, and then you take a lamp and you directly shine onto the skin. This is kind of straightforward. However, there are many tumors inside the body, but people have really been busy developing techniques for shining light into the body. Here is for brain cancer example where they really put a balloon inside the cavity where the tumor used to be, and the laser is connected to the balloon. The balloon is scattering light, so to make sure that the light is distributed very homogeneously. It's quite amazing to see. There's been also clinical trial for pancreas tumor, for which we have no current treatment available. This is an example from the Dutch Cancer Institute in Amsterdam, where they treated a tongue tumor by putting some optical fibers over there. And you can see very well how to bring the light into the tumor. The end of the optical fiber is terminated by a light diffuser, which is inserted into a syringe. The syringe goes into the tumor, and then you can get a lot of light actually in a tumor. So of course, I'm not doing this, but this all exists, and it's important to realize you can actually shine light almost everywhere in the body. And before I go further, I wanted to spend 1 minute on the how to measure actually what is a light-activated drug in vitro, because as a chemist, I'm going to do very little work in patient, at least for now. So we are mostly testing our compounds on cells. So basically, you have 96 well plate. If you want to test, for example, 3 drugs, you're going to have to use 2 plates. So one plate is going to be called the dark plate. It stays in the dark, and you basically, for each of the drug here, you make a dose response curve in the dark. Then the second plate is the light plate. So here's green light, so I put it green. You repeat the same thing, but now you shine light on the cells in presence of the compound for a certain time at a certain dose of light. And then you get a second dose response curve. Here is concentration, and here is relative cell population compared to non-treated wells, let's say. And you get what's called efficacious concentration, EC50. Sometimes people call it IC50. So it's the concentration needed to kill 50% of the cells compared to non-treated drug. You get a value for the dark, you get a value for the light. And if your drug is light activated, then your EC50 goes down by a certain factor. And this factor is called the photo index, EC50 in the dark divided by EC50 in the light. And basically, we try to maximize the photo index in different conditions with the compounds we're making. So I prepare a small history of phototherapy with metal complexes just to put my work in perspective. I will try not to spend too much time here. I would like to say that PDT has started a long time ago, like thousands of years ago. But the modern history of PDT, let's say, can be related to the 50s, where there were still very few treatment possibilities for cancer patients. And people realized that when you put some porphyrins in tumor patients, you get some fluorescence into the tumor. A bit later on, Doherty realized that if you shine light on this tumor, the fluorescence shows the porphyrin is here, and if you shine light on it, then you will get an anti-cancer effect. And that was, of course, the big discovery. Another big discovery done a couple of years after the initial efficacy discovery was that the main agent was the singlet oxygen. And this has led to huge, developments up to, let's say, 95, where the first photosynthesizer was approved for esophagus cancer, actually. And then the PDT history has gone forward, and I mean, it's been in huge developments also in the last decades. But I did want to switch here to the metal-based light-activated compounds. And to me, it started with Morrison in 89, 92, who was studying rhodium complexes, and he demonstrated the idea of what he called the photo-cisplatin. Now, cisplatin binds to DNA. He said, is it possible to activate a metal compound that would bind to DNA like cisplatin, but only after light irradiation? And he demonstrated this with rhodium-3 compounds that were only absorbing UV light. So this idea has been probably lying around, and Patrick Bednarski and Peter Sadler developed the first platinum-based prodrugs, light-activated, and they had to go to platinum-4 because platinum-2 don't have a lot of photochemistry. And this was further developed, I think, with Claudia Turo and Kim Dunbar, who worked further with rhodium-2 dimers. And then the ruthenium chemistry started again with Turo here. I started my career more or less over there. And in 2002, there was a very important paper from Phoebe Glaser about the binding of ruthenium polypyridyl complexes to DNA after visible light irradiation. And of course, since then, people have been trying to push the light towards the red. So the first part of my talk will be dedicated to something which Peter Sadler introduced first as photoactivated chemotherapy. So they published all this naming in a Dalton paper from 2009. And the idea is basically that of these photoactivated cisplatin. So you take a metal complex, you make sure that the metal complex is saturated with ligands, so it cannot bind to DNA. It is a prodrug. It is not toxic because it does not bind to DNA. And the idea is, okay, if you shine light and your ligand is kicked away, then your ruthenium recovers the ability of aqua complexes to bind to DNA, and then you will kill the cancer cells. So what I realized when I started is that it was not necessarily, well, very nice to just make another molecule like which is doing something which PDT is doing as well, because PDT is much more advanced than PACT in the clinic. So what I tried to really think was what is the added value of such a principle? And I realized that in principle, this photosubstitution reaction, they work very well under hypoxia, so in absence of oxygen. They don't need oxygen to work, while PDT inherently relies on oxygen activation. So then I oriented my research towards trying to demonstrate that PACT can be used in oxygen-poor conditions. And now is the question, well, how do you do that in details? Yeah? So, of course, PDT works a lot. There are also cases where PDT doesn't work. And so a friend of mine, so Michael Heger from the Amsterdam Medical Center at the time, he made a review on the clinical outcome of PDT, and he realized that for, depending a bit on the conditions, there were a lot of patients that responded very well to PDT, and there were other patients that did not respond at all, or very badly. So you see here 25% of skin cancer. And for some form of cancer, like biliary cancer, really it doesn't work. I also wanted to mention another study from Teresa Bush, much more recent. This is on tongue cancer patients and mouth, well, floor of mouth tumors. What they did is they split patients. They came with these type of tumors as a function of the oxygen concentration in a tumor. Then they treated them with PDT, and they looked at the response. And here as well, well, some PDT respond very well, some others don't. And when they looked at it back after the study, they realized that the people who did not respond to PDT at the beginning, when they entered the study, had a very low oxygen concentration in the tumor, while the ones that had a very good response had a much higher oxygen concentration in the tumor. So this is kind of logical for PDT, which is based on oxygen activation in oxygen. So if the tumor is oxygen poor, it doesn't work so well. But the quite amazing thing is that this hypoxia problem is actually a general problem in oncology. So this is a different kind of study. It's a study about cervix patients. And then it's all treatment mix. So it's not specifically PDT, but basically they also split the patients as a function of the partial pressure of oxygen in a tumor when they entered, well, just after diagnosis. And if you look at the survival, you see that the survival when the pressure of oxygen is high enough in the tumor is much better than when the pressure is low, which means that hypoxia is a problem not only to PDT, but also to many other forms of treatment of cancer, like radiation therapy, for example, but also chemotherapy, even surgery and immunotherapy. So it's kind of a general issue that we are tackling, and this is also why it's interesting. So what is this idea of hypoxia in cancer? Well, the idea is that in a tumor, you always have regions that are very close to a blood vessel, and these regions are going to be quite well oxygenated. And then you have regions that are very far from them, and usually the cells are dead because they can't really live without oxygen. But there is also a region where the cells are hypoxic, so the oxygen concentration is too low for the cells to really, well, to be like normal cancer cells, so to say. But it's also too high for the cells to die, and they will recur later on. They are the difficult guys that we try to kill. In vitro, it's kind of easy to demonstrate that PDT doesn't work without oxygen. This was a small study we did on lung cancer with rose bengal, which is a typical PDT dye. You see in the dark, there is no toxicity. In the light, you see toxicity due to the PDT effect if you shine light in air. And if you go into hypoxic conditions of 1% of oxygen, then you see that your cytotoxicity of the rose Bengal with light irradiation is completely canceled because there's not enough oxygen. So what we demonstrated in 2017 is that if you take a PACT complex, so this was our first study on hypoxia. This is a ruthenium complex you see with a slightly, well, this is a quinoline ligand instead of a BP, and the cytotoxic thing here is the ligand. So this ligand STF31 is actually a cytotoxic inhibitor. And so in this, well, in this form, it is caged by the ruthenium, so it's not so cytotoxic. You see the normoxia, so the data here on the left, there are in vitro data. These are the EC50 directly reported on the graph for lung cancer and skin cancer. So you see the EC50 is higher in the dark than in the light. You see the light activation. This is normoxia data with a photo index of 2 to 3. And when you go to hypoxia, you shine light, and what you see is that you keep the light activation, so a higher cytotoxicity in the dark compared to the light. Sorry, a higher cytotoxicity in the light compared to the dark. What was actually quite amazing is to see that the photo indexes were almost equal under hypoxia and under normoxia. So this was the first demonstration that indeed PACT still, the principle of PACT still works in a hypoxic cell. What you might realize is that all these values are higher in the hypoxic data set than in a normoxic data set. And this is something we are still facing today. So this is for another compound. It doesn't really matter. You see here the dose response curve in the dark and in the light of a specific compound and the normoxia. You take the same compound, the same cancer cells, and you go under hypoxia. You see that the EC50 in the dark is shifted to the right. The EC50 in the light is also shifted to the right. So this basically means that the cells are more resistant to chemotherapy in general. And there are all kinds of reasons. I'm not going to do a whole course on hypoxia today, but basically there's a first, well, a master gene of hypoxia basically that is switched on when the cell feels a lack of oxygen. And these gene expression, well, leads to a lot of different mechanisms to change, to survive the hypoxic conditions, to change the mechanism by which they take their energy, to overexpress efflux pumps that are kicking away all the chemo you're trying to give to these cells. So that they are on average more resistant to chemo. So this is quite a challenge to treat this kind of cells. You can see here the same cell treated. So these are skin cancer cell treated under normoxia or the hypoxia. And here we highlighted different proteins. So cadherin, fibronectin, vimentin, and tubulin and actin. And what you can see is that the cell completely look, well, the protein expression really looks different if you're under normoxia and hypoxia. So basically, any cancer cells in the normoxia and hypoxia are different beasts. They change by the hypoxia conditions, and they become more difficult to kill. So let's have a step back to chemistry. So if you take a ruthenium compound and you shine light on it, you can have a lot of things that happen, but you should never forget that in most cases, the excited state just goes down to the ground state and nothing happens. So there are molecules that just are colored and that don't do anything from the excited state. Now there are other molecules which have good phosphorescence, and they are used for looking at cancer, for example, so photodynamic diagnosis. What you can do is, as we discussed before, you can generate a singlet oxygen by energy transfer from the excited state. This is called PDT type 2. Now there's another form of PDT which is called PDT type 1, which consists in transfer of electron instead of transferring energy to oxygen. And then you generate superoxide, which is also cytotoxic. And the one we are mostly interested here is PACT, so where you do the photosubstitution. Now, all compounds do not photosubstitution. So if I come back to this ruthenium terpy BP thioether complex, it does so because it has these low-lying pi star orbitals that are located on the polypyridine ligands. They're actually a bit lower than the EG orbital on the metal. So when you shine light, usually you get what's called an MLCT state, so metal to ligand charge transfer, because the electron is going from a metal-based orbital to a ligand-based orbital. Then you get a spin flip because you have ruthenium, which is a heavy atom. So you get mostly, very quickly, you get a triplet state. And for certain type of complexes, then these EG orbitals are not too high in energy. So with thermal energy, you can promote this electron here to the EG orbital, and then your excited state is called a metal-centered state. So it's a triplet metal-centered state. And these orbitals are anti-bonding, as you know. So this leads to the ligand dissociation by one of the solvent molecules. By the molecules in the solvent. So you need a low-lying MC state to get photosubstitution running. So we can have a look at this in terms of an energy scheme. It's quite important because you have your ground state here. You go to the singlet MLCT, which transform into triplet MLCT. Usually these excited states, they are responsible both for the phosphorescence and also for singlet oxygen generation for PDT mechanism. And so you will have these things mostly when the MC states, they are high in energy. And if your MC states are much lower, and sometimes they're even lower than the MLCT state, then it's easy to get to them and to get to the photosubstitution. What is important to realize is that if your molecule is a good PSCT molecule, usually it's a bad PDT molecule because then it cannot do, well, let's say the competition to get to the MC and the photosubstitution is in favor of the photosubstitution, and there is not so much phosphorescence and not so much PDT effect. So I can exemplify this on the reaction I've been showing to you for a while. You see here, I told you already. So we quantify the quantum yield here. We have 2% of photosubstitution for that molecule. And what you can see is that the quantum yield for singlet oxygen is very low, and so it's less than 1%. It's hardly measurable. So this is really not going to do any PDT, this molecule. And the phosphorescence quantum yield is also very low, less to 10⁻⁴. It's at the limit of being detected, and you certainly cannot see it by eye. Good. So now you know most of the things about how to design a PSCT. Still, there are a couple of details to do them properly. So I'm going to go into a few of the details. One of the questions is whether when you make a molecule that splits in two, who is responsible for the toxicity? So sometimes it's the metal part, sometimes it's the ligand that you kick out. You have synthesis issue. Obviously, you need to be able to make some of these compounds. There is a hydrophobicity issue when you make metal, well, when you make drugs in general. Then there is all the photophysics. So do you get more substitution or is it photostable? Do you get any PDT effect? Because if you kill cells with light, you always have to verify whether you're doing PDT or not. Then what is the color of the light? I'm not going to necessarily to activate your molecule. I'm not going to discuss that too much today because I can discuss everything. Once you've set up the chemistry running, then you put them in cells. And then, of course, it becomes much more complicated. The first box to tick is there any activation in an homoxic cell? Then you go into hypoxia. Then you try to see how it's working. And what I'm going to discuss with you a bit today is how to get, how to try to go towards in vivo. Of course, we're chemists, but we would like to go one day to patients. And so we need to understand what's going on when you put a drug in an animal. So the first small warning for all people interested in light-activated drug is it's important to realize there are molecules that are chemically activated, like this one. You have 2 BPs on the ruthenium and this NS ligand, which are called MTMP. It's a pyridyl thioether. When you shine light on this molecule, you have a very selective photoreaction. You kick out the bidentate ligand here. You get the bis-aqua complex, which you could say it should bind to DNA. You can characterize all these things. So the quantum yield in blue light is 0.3%. It's a bit low, but it's perfectly fine. But if you take this molecule and you put it in a cancer cell, so this one is, again, is a lung cancer cell, which you use as a kind of reference, then you see that there is absolutely no effect. Basically, the cells don't care whether or not the photosubstitution has happened or not. We were a bit surprised because in principle, we were thinking that this bis-aqua species should be phototoxic. It should bind to DNA. All the more that, a couple of years ago, some years ago, Phoebe Glaser published this compound, which is a sterically hindered ruthenium complex with a dimethyl BP instead of the NS ligand we are using. So when you shine light on this molecule, you kick away the dimethyl BP ligand, you get the same bis-aqua ruthenium complex. And if you put that complex in cancer cells, you see in the dark, there's no cytotoxicity. And in the light, you get a very nice EC50 around 1 macromolar, which is very nice. So we could reproduce, before I come there, yeah. And what Phoebe also showed is that if you take DNA and you mix it with this molecule, in the dark, there is no interaction. And if you shine light, well, the more light you put and the more complex, the more your DNA seem to be modified compared to the dark, basically because this molecule indeed binds very well to DNA. Now, we tried to reproduce these results and we got very nice activation. Here's the dark toxicity, the light toxicity. So that was not a big deal. So something else was going on. And after investigating, we realized that actually the main difference between Phoebe's compound and our compound was not really the hydrophobicity or so. It's very similar between both complexes. These are both water soluble molecule, basically. Their uptake, when you put them in the same cell, in same conditions, is very similar. I see 1.3 or 1.3, basically, nanogram of ruthenium per million cell. So which is very low. So it basically means these molecules actually don't enter into the cell before light activation. But what does happen is that we generate this NS ligand, which is non-toxic, both in the dark and in the blue light irradiation. While the dimethyl BP was strongly cytotoxic, which was a bit unexpected, but it does happen with IC50 is around 5 macromolar. It depends a bit on the cell. So we believe this complex is actually phototoxic, not because of the ruthenium bis aqua, but because of the dimethyl BP. So then the question is, can we actually make a metal-based complex where the phototoxicity is due to the metal, the bis aqua species that you generate? So it's okay, well, if the hydrophobicity is a problem, because these two molecules were not going into cell, let's improve the hydrophobicity. So we changed the BP into diphenyl phenanthroline, which are super greasy. And we generated the same, well, the derivative of the previous molecule. Now you see the log P has become positive. So indeed, this molecule is still water-soluble, but much more hydrophobic. It is still photoactive. So when you shine blue light, you get a bit lower quantum yield, 0.1%, but still the reaction is perfectly fine. And when you put it into the same cancer cells, then we see a nice activation indeed. Now we know that, okay, the NS ligand is kicked out. We know the NS ligand is not toxic. So now that we see, and we know, so it's not a PDT compound. So now we see that this activation is really due to the ruthenium fragment, this bis aqua complex we're generating by photosubstitution. The photo index is kind of okay. It's not very high, kind of okay. But what is a little bit problematic is that the dark cytotoxicity is actually, well, around 10 to 20 macromolar. So it's quite low, which means the compound in the dark is already quite cytotoxic. So of course, you get more cytotoxicity after light irradiation, but the dark toxicity is too high for something which is not supposed to do any harm. So then we went back into the paper and we designed another complex. We took kind of the mixture between both. We keep one diphenyl phenanthroline and we replace one by BP. When you look at this molecule as a coordination chemist, you should realize that this is a triceteroleptic compound with 3 different bidentate ligands. So it's a bit more tricky to synthesize. And when you try the regular synthesis route, you get ligand scrambling. And so you get 2 NS ligands or 2 BPs or 2 diphenyl phenanthroline. So we had to take the method that has been developed by Tia Case in Dublin, where you introduce first the first ligand. This is going fine. But in the second step, you see we're introducing, well, she proposed to introduce this oxalate ligand. Because oxalate is charged, while all the other ligands are neutral. Now this molecule can be isolated quite well, and 2 oxalates are not going to bind to that fragment. Once you isolate this, you add acid, you kick away the oxalate, then you add 1 equivalent of BP, and you can isolate that molecule very nicely. So once the molecule was available, then we checked the photochemistry here as well. No big deal. The NS ligand is photosubstituted the same way as the 2 other molecules I've shown to you. Here is an acetonitrile. Because it's in acetonitrile, the quantum yield is a bit higher. Then it was very exciting to see what happens in a cell. We saw a very nice activation in A549 cancer cells with a much higher photo index of 10 instead of 5. And basically, if you compare to the molecules we just discussed before, so of course the light was not the same because it has not been done the same way. But if you consider just the dark toxicity curve, you see that the heteroleptic molecule is much less hydrophobic. So it has a lower toxicity in the dark compared to this one. And we lose a little bit in phototoxicity, but overall, we gain the ratio of the dark versus the light toxicity is in our favor, and we have very nice light activation here. If you look into the uptake, so we treated the cell at the IC50, and you measure the uptake by ICP-MS, and you see here we got the same number of ruthenium per milligram protein here, but as one is much more toxic in the dark than the other, you see that the most cytotoxic was treated at 3 micromolar, while our retroleptic was treated at 65. So meaning you need, well, 20 times more of that molecule to get, let's say, these 2 nanomole ruthenium per milligram protein into the cell, which means basically that this molecule is just, well, much more right in terms of hydrophobicity and hydrophilicity balance, while the other one is clearly too hydrophobic. So we were quite exciting with this molecule. So we started to look at other cancer cells. We went into eye cancer because I have a project on eye cancer. So as you can see on conjunctive melanoma cell lines here, MMM1 and 2, the activation is going quite well. We also went to metastatic prostate cancer cells, and there really the ratio is 10 times higher, which is really nice. So we went to a colleague in Leiden, Eva Snar-Jagalska from the Biology Institute. She's a specialist of the zebrafish embryo model. Tu t'en vas, s'il te plaît. Merci. This was yes, that would be it. Sorry for that. So Eva Snar is a specialist of the zebrafish embryo model, so which has been used quite a lot to test anti-cancer compounds. Because they are small, you can have good statistics, much better than with mice. They look quite a lot like humans, although it might look a bit surprising to some of you. There are many genes that are in common. Also, they are transparent, so it's quite easy to look at everything that's going on in this embryo. The green here is the vasculature, and the red is typically the tumor cells. So by using transgenic tumor cells, you can really look directly at your tumor burden. So the protocols we published in Cancer very recently, basically 2 days post-fertilization. So this DPF is day post-fertilization. The embryo were injected with a tumor burden. So you take 200 cells, you put them into the fish, then you wait for 24 hours and you treat. Yeah. We had no idea where to go. So we tried 3 different treatments. One was to add the compound inside the water in which the embryo is swimming. This is called water administration. Then we left 12 hours a drug to light interval to let the compound get into the animal. Then we refresh the water and we shine light. And then we had intravenous and retroorbital injection. So this is directly, you take a tiny volume of a solution of the retaining compound and you inject it directly. So either in the dorsal vein of the animal or behind the eye, because we were working on the eye cancer. And then we had a much shorter drug to light interval, because obviously you don't need so much time for the drug to travel around the body of such a small embryo. We had to repeat this 4 days in a row, and the irradiation was done for 90 minutes, which corresponds every time we do it to 19 joule per square centimeter. We did it 4 times, and that was green light irradiation. And as I said, we quantified the tumor burden by microscopy. You could look at total fluorescence or the fluorescence area. So of course, we first tried the prostate cancer because the PI was so high. And so first, the most easy way to do that is to put it into water. But as you can see, in terms of quantification, or also in the photo, there is no significant difference in terms of tumor burden between the let's say the ones that received the compound and the ones that just had the MSO injection. So it was kind of disappointing. One should realize that when you add prostate cancer cell into zebrafish by injection, the prostate cancer cell go into the tail. So then it's called an ectopic model. An ectopic model is when the xenograft is not in the tissue it comes from. It's not a prostate tissue. So this didn't work. We tried another ectopic model where this time you inject the compound directly into the circulation. The cells were injected in the same way as before, But here as well, you see no significant effect for that ectopic model. There is no effect over there. However, when we went to an orthotopic model, orthotopic means that the tumor xenograft is actually in the kind of tissue where it comes from. So obviously it's a human cancer cell, and this is zebrafish. So it's not the same, but it's an eye, let's say, tumor into, well, which is injected close into the eye. You can see here the tumor that has grown for the vehicle control. In the dark and light, we see no effect. In the dark with the compound, we see no effect, but you can kind of clearly see on the photo that when you combine the ruthenium and the light, the tumor burden has gone down. And if you quantify that, you see that the decrease of the tumor burden is quite significant. So we were very exciting here, or we are actually very exciting, because this is a direct proof that we have activation of our PST compound in vivo. And there is not so much such studies. What I wanted to highlight is that the photo index in vitro for that particular compound in this particular cancer cell is actually lower than the one in the prostate. So it's quite important to realize, and that's one of my main, home message for today, is that a high PI in vitro is not necessarily a reason why a compound is going to work in vivo. If a compound never reaches the tumor in the animal, then nothing is going to happen. So obviously here, the orthotopic model, that was much better than in the ectopic model. Yeah, this is something I learned personally. And I also wanted to mention that obviously for zebrafish, it's kind of challenging to make local activation of the drug. You activate the compound in the full body, which is a small disadvantage of that model, but it's not so much of a big deal. You just look at the on-on effect of the photosubstitution reaction. And to finish with, I wanted to discuss another work which is quite recent, which we published last year. Coming back to the case where these ruthenium complex is actually not cytotoxic. We already met one. So when the ruthenium complex is not cytotoxic, you can say, well, we can throw it to the bin. But we try to, to think another way. We say, well, maybe if the ligand would be cytotoxic, then we would have the same effect, because then you say now the ruthenium is preventing the ligand to exert its activity in the dark. So this complex is not toxic. And now, instead of being the ruthenium that goes to DNA or to somewhere, then it's a ligand that, for example, would be a protein inhibitor, and that would bind to a protein. And this would not happen in the dark. So this is something called photocaging, because in the dark, the cytotoxic ligand is caged by ruthenium. And after you shine light, you do photosubstitution and you uncage the ligand, and you release, obviously, the caging group as well. But if it's not cytotoxic, you are going mostly to see the effect of the ligand. So this field of research is very active right now. There's been quite some work from different kind of groups. And I wanted to, discuss the work I did with Alexander Kornienko, because we did go to in vivo studies with this molecule. So Alexander Kornienko is from Texas State University, and he's working with microtubule polymerization inhibitors, which are called MTI here. So these are cytotoxic compounds which bind to the colchicine sites of the tubulin protein. And tubulin is a protein that makes one of the two main types of cytoskeleton in the cell. And one of the compounds he published had these thioether. So when we met, where we said, okay, it would be quite straightforward in principle to coordinate that thioether to the ruthenium and to cage the MTI molecule. So indeed, it's quite straightforward to do that. You have some hydrophobicity issues to solve, but it is quite fine. And when you shine light on the cage compound, so here's green light, you recover your caging aqua complex, which is non-toxic. We know it's non-toxic. And a very cytotoxic MTI. The quantum yield is around 0.5%, which is around the values that we like to have. So the photochemistry of these things goes well. It is not emissive. It does not do anything with oxygen. And when you put it into a microtubule polymerization assay, you see that it works very well. So a polymerization assay here, you get a lot of fluorescence when the microtubule polymerize. So paclitaxel is something that blocks the polymerized microtubule, and you can see here we have high fluorescence. When you get either the control or the ruthenium MTI in the dark, so that's the gray curve here, you see that this is the native, let's say, rate of polymerization. You have normal microtubule formation. And when you shine either blue or green light over there, you block the polymerization. So your microtubule remains depolymerized. So this is well, the proof that the molecule is doing what we want. Now we wanted to see that in a cell, and we went to the Leiden Academic Center for Drug Research in Leiden, where I have a colleague, Sylvia Ledevedek, who is an expert in bioimaging. So what we can do here is image the nuclei, the microtubule, and the actin at the same time. And when you do that with a DMSO vehicle control, you see that, well, nothing happens whether you shine light or not, and the light itself is not toxic to the cells. However, if you take 10 micromolar of the molecule, you see that in the dark, the image really looks like the vehicle control, but in the light, you have a massive change. The microtubule seems to be quite strongly, well, to have some problems. And if you put a bit more of the molecule, you see that there is almost no polymerized microtubule anymore in the cells, which is obviously going to pose a huge problem to the further development of the cancer cell. So in terms of cytotoxicity, these are the dose response curve of the cage. As I told you, it's not cytotoxic, this ruthenium aqua complex. The MTI cage itself, so the caged complex is, well, it has a cytotoxicity between 10 and 20 macromolar, depending on the cell type. When you shine light, you move it down to below macromolar, and you get the green curve, which is very close to the free MTI inhibitor, which is here. So in terms of values, you see that in skin cancer cell, either melanoma or non-melanoma, we have more than 20 photo index. In FR49, we have 4. And then we went to hypoxia. So in hypoxia, well, it was actually impossible to get the IC50 over there, but you see that the photo index on the hypoxic cells of the same type is more than 12 and more than 16 here. So basically, we keep the strong photoactivation of this PICT molecule under hypoxia. And here where we do have a value, you see that actually the value is like the first compound I've shown to you, almost exactly the same, which is also a very good, well, sign that the PICT is working on a hypoxia. And in a PDT conference, which happened in Nancy some years ago, I met these two gentlemen who are doing PDT in vivo in mice. And it was a very nice meeting. And we decided to work together. So I sent them the compound, and they tested our compound on a real mice model this time. So this is a lung cancer xenograft in a nude mice, so without immune system. The light, well, the dosage, we had no idea where to start from. And we did 1 milligram per kilo intraperitoneal, and that was fine. There was no toxicity in the dark. We used 24-hour light interval at 40 milliwatt, 10 minutes twice. And what you can see here on the tumor development is that the compound, which is not irradiated, is very comparable to the curve of the, let's say, control, well, the mice that have received a control, vehicle control. The only curve that goes away from these bunch of curves over there is the ruthenium complex, which has been irradiated, where we see a modest 30% reduction of the tumor volume. But considering that we had no idea where to start from, and this has been absolutely not optimized, we were quite exciting to see that we could see an anti-tumor effect in vivo in a mice using PICT. So I think this is the first time this has been shown, and we got a nice JACS paper last year. So it will be time to close. So I think if you remember from my talk that you can activate an anti-cancer compound by a photosubstitution reaction that liberates, well, that splits basically a ruthenium complex into two parts, the metal-containing one and the ligand part, that would be good. Obviously, this is designed for hypoxia. This is a bit retrospective view on it, but let's say it also works on the hypoxia, which does not mean that it does not work in air. It perfectly works in air. Quantum yield are the same. And, but because it also works in the hypoxia, we try to develop this technology in hypoxic cancer cell first, and we are trying to go to hypoxic tumors or tumors which have a high volume of hypoxia inside the tumor volume, so to say. Well, I'm quite persuaded that we need a lot more in vivo data for these kind of things, that this is one of the reasons why the compounds that are now either approved or in clinical trials have been successful, is that they started quite early on getting in vivo data. So we are working on this. Still, it's quite challenging for chemists to understand what's going on in vivo and the different types of model. And I really encourage everybody to work with biologists and well, consider these details that really matter. I would like to finish my talk by thanking my group. They are really awesome people. I know they are listening to me at Leiden in a room in common. So hello, guys. And so the work I've shown to you was possible because Bianca Sievert and Samantha Hopkins, two postdocs who now left my group, but they really introduced the biology in my group, and now it's running very well. So the work I've shown to you today is essentially from Jordi and Vincent, also partly from Lucien Lameyer, who is here. I would like to thank the funding, without whom all this would not have been possible, so ESC and NWO, for the support to my group. And I also wish to mention that I am looking for a postdoc. And last but not least, we were supposed to have a conference in Amsterdam this year, a photochemistry conference from EuroBIC. I was in the organizing committee, so I know quite well the experience from Ziga in Iceland. And we were lucky this time that we could shift the whole series of conferences of 2 years. So the EuroBIC photochemistry conference is going to take place in 2022. So you can mark your agendas. Thanks again for the invitation, and I will be very happy if you have any questions I can answer for now.
Well, thank you very much, Sylvestre, for an excellent presentation. I'm sure we have some questions, and now we have to figure out how we get the questions. Let's see. So we have, here we have, so we have a few comments.
Yeah.
Excellent talk, and thank you for a very nice talk, very exciting from the audience here. But do we have any questions? I have to say that I was very excited. This may be not directly related to your results, but I was really excited to see the inside of an esophagus. And I saw very clearly how that healed very well.
Yeah,
it was a very nice demonstration of the PDT working. And I think also it's extremely interesting to see how the normoxia and hypoxia have played together in this scenarios with the molecules, either with a toxic as a molecule or a ligand toxic situation. Anyone that wants to ask a question, just raise their hand.
Carlo has a question.
Yes.
Hi, yeah, great talk. It was really great to hear all the recent results from your lab. So one thing I was wondering, so when you lose the ligand upon photoexcitation, is there any chance that the complex undergoes any isomerization? Have you looked into that possibility? And if the isomerization can have any impact on the cytotoxicity?
So yes, definitively photoisomerization does happen. So for example, it's a reason why when you work in particular with ruthenium tris-BP kind of complexes, which as you know are chiral, most of the time when you have photosubstitution on this family of complexes, then you always lose the chirality. So it's in principle not useful, let's say, in biological terms to test the phototoxicity of both isomers, because you know whatever you start from, you're going to have the same. Which doesn't mean the isomers would not have a different uptake, for example, or dark toxicity. So the dark toxicity can be different between isomers. And I think we have measured, oh no, that was, yeah, that was an example where the ligand was chiral, and we tested both enantiomer of the ligand, but bound to an achiral ruthenium. And then we saw a difference of dark toxicity for both isomers. However, we never saw a difference of phototoxicity, so after light irradiation, because of the reason I just said. There are also ruthenium compounds that isomerize not in terms of, let's say, making the other enantiomer, but really like TerpB, for example, they can rotate like this. And these, formally, you make diastereo isomer. So in principle, it would be possible, but it's a bit difficult to study because you generate one isomer or the other inside the cell, which is usually different from starting from, let's say, hi, nice to see you, which is different from starting from one or the other enantiomer outside of the cell and hoping they are going to get into the cell the same way as when they enter as a protected drug, and then you generate the isomer inside by light irradiation. So it is a bit tricky to study, let's say. But yes, photosubstitution often leads to photoisomerization.
Yeah, because for your pyridine thioether ligand, when it goes out and you don't see any cytotoxicity, you said that it's because of the co-ligand which is coming out. It's non-toxic, but it can also happen that the paper that you were comparing it with, in the two cases, you generate two different isomers, and one is cytotoxic and another is not. I mean, how do you know it's actually the co-ligand which is causing the difference in the cytotoxicity?
I'm not completely sure which work you refer to. The TerpB thioether doesn't have two isomers, yeah? Yeah, it has only one.
No. I mean, yeah, you were talking about the ruthenium diaqua complex, right?
Oh, the bis aqua, yes, the bis aqua, yeah.
And initially in the Jack's paper from a different group, I don't remember, they proposed it's the ruthenium compound which is responsible, the bis aqua. And
yeah,
then
the bis aqua,
yeah, so it can there be two different isomers? One is active, one is not. And in the two cases, you actually generate two different isomers.
You're right. So in this particular case, so when you start to kick away a bidentate ligand, you first get the cis bis aqua, and then you get the trans bis aqua. So this one, we see it. And we have studied also the compound published by Phoebe Glaser. So we see very clearly the trans bis aqua being formed. So I am not aware of the difference between the cis and the trans aqua. This, I don't know how they compare. Also in terms of DNA binding, in principle, they should be able to bind both, but they will do different DNA adducts. So yes, that would be an interesting thing to study, I think. Yeah.
Thank you very much. Great talk again.
Thanks a lot. So I see, what is your thought on multi-metallic system for PACT or PDT? From Ashish Patra. Hello, Ashish. Aziz, yeah. Well, I'm sure it can be developed. I actually had a project on multi-metallic at some point, but we never went too far. The basic idea was to get two ruthenium compounds which can bind each to one ligand, let's say. So two ruthenium TerpB, and to bind them together, and to hope that when they interact with DNA, they would form, let's say, inter-strand DNA adducts. So I do think there is a future for this, but we are not investigating this right now. So what happens when both ruthenium complexes and, well, the ruthenium complex and the ligand are toxic? This is a question from Marco Chino. Thanks for your question. This is a very good question. We are really much into this. So that's, I think, part of the nice thing with PACT is that you can combine the action of two drugs and that they will hide each other. So we are working into this. Certainly, there are a couple of papers already from Claudia Turo, also maybe from Phoebe Glaser, I would have to check, where they combine PDT with the action of a well, let's say, singlet oxygen generation with the action of the ruthenium complex. So yes, there is a lot of opportunities here to develop stuff and to try to make synergy. However, getting synergy is not necessarily easy. So we are working into it, but it's not an easy thing. either additive or yeah, that go against each other. Both could happen. So you have to do it properly. Yeah, that was clear. Did I answer your question, Marco?